Welcome to the presentation of our CVPR 2026 highlight paper, Unsupervised Multi-Scale Segmentation of 3D Subcellular World with Stable Diffusion Foundation Model. In this work, we leverage the internal learning mechanism of the Stable Diffusion Foundation Model to segment subcellular objects across multiple scales from extremely noisy and crowded in situ 3D microscopy images of cell. Current vision foundation models has become highly effective in understanding the visual world around us. However, can we say the same about the world underneath the human cell? Let's start by discussing how to even visualize the extremely complex 3D subcellular world in their native state. With emerging imaging technologies like cellular cryo-electron tomography or cryo-ET, it is now possible to image the 3D subcellular objects inside the cell in their native environment. In this technique, a cellular specimen is rapidly frozen and bombarded with focused ion beam to ensure appropriate thickness of the sample. The specimen is then imaged using an electron microscope from multiple tilt angles as shown in this figure. The tilt series images are reconstructed into a full 3D tomogram volume that contains the visualization of 3D subcellular world up to sub-nanometer resolution. However, obtaining useful information on the subcellular world from such cellular cryo-ET images is not a straightforward process and is hindered by multiple challenges, primarily driven by the nature of the images. The images are extremely large volumes with very low signal-to-noise ratio and densely crowded with subcellular objects. Furthermore, the subcellular objects inside the cell widely varies in shape and size, requiring segmentation across multiple scales. All these makes manual annotation expensive and burdensome. Furthermore, these volumetric images have high intra-domain variability. For instance, a tomogram of bacteria cell is highly different from a human cell. Even images of same cell across different biochemical environment is very different. So even after burdensome annotation on a set of these images and training a supervised model using the annotations does not help much in generalization. In this work, we investigated if we can solve these issues with an unsupervised solution that does not depend on expensive manual annotation, is effective for subcellular objects across diverse scales and sizes, and can be used robustly across multiple domains. To this end, there were two options. First, there exist data-driven traditional methods like ChanVs and watershed segmentation. However, they were designed for high SNR images and performs poorly in low SNR cellular tomograms. Another option is to perform zero-shot inference using supervised foundation models. However, they were trained mostly on natural images and is not effective for cellular tomograms yet. So, in this work, we took a new direction. We took advantage of the fact that objectness pattern emerges within attention layers of vision foundation models. For instance, here we can see objectness patterns highlighted in the final layer attention map of several vision foundation models, where Stable Diffusion Foundation Model provided the most detailed and clear objectness pattern among others. A recent study found that the query key features of the vision foundation models capture where pathway of the data, useful for segmentation. Consequently, in this work, we used query key features of the Stable Diffusion Foundation Model to perform unsupervised segmentation of cellular tomogram images. Now I'll walk you through our method step by step. We ask the user to pick a full 3D tomogram and select a small range of slabs. These are 2D slices along the z-axis that are visually rich in subcellular structures. For example, in our S-Pombe experiments, we used around 100 slices from one representative tomogram, which is only about 1 to 1.5% of all slices in the dataset. We then do simple pre-processing, like contrast stretching and enhancement on these slabs. Since each of these slabs are too large for a vision foundation model, we split each slab into 4 non-overlapping patches. These quarter images are our unsupervised training data. Here comes the core part of our method. We use Stable Diffusion Foundation Model to obtain the eigenvector features necessary for segmenting our training data, or the quarter images. To this end, we fed the quarter images to the pre-trained Stable Diffusion Model, extract features generated from query key matrices, concatenate these features across all attention layer, and form the multi-layer affinity matrix. Then we perform gradient descent-based spectral clustering to obtain the eigenvectors of the affinity matrix. We typically select top 12 eigenvectors and reshape them to eigenvector images. We developed a heuristic called diversity score that measures the patch area-wise pixel diversity in the eigenvector images. High diversity scores typically correspond to important features, whereas low diversity score means mostly background and noise. We select the best eigenvector image based on the diversity score and enhance it by iteratively aggregating additional eigenvector images into it until the overall diversity score of the eigenvector image increases. The resultant image serves as the feature image, which we further correct using information from neighboring slabs across depth axis. We perform Gaussian adaptive thresholding to the corrected feature image and obtain the mask of our quarter image patches. This mask contains both large membranes and small macromolecular objects, which appear as small repetitive patterns across the mask, similar to how cells look like in tissue images. Due to shear size differences, the large membranes and small macromolecules need to be differently processed in downstream. Hence, we used a pre-trained cell segmentation foundation model as an oracle to separate macromolecules and membranes from the obtained multiscale masks. By combining the membrane mask for the quarter images, We obtain the full membrane mask for each selected slab of the tomogram. For faster inference and mask refinement, we train a supervised U-net regarding these unsupervised masks as pseudo-ground truth for the selected slabs. Then we use the pre-trained U-net to infer mask across all slabs for all tomograms in the dataset. On the other hand, for macromolecules, we extract the locations of them for the selected slabs and train a supervised particle picking model regarding these locations as pseudo-ground truth. We then use the trained particle picking model to infer macromolecule locations for all tomograms in the dataset. We evaluated our pipeline against cellular cryo-ET dataset of S-POMB. Here we show the qualitative results on membrane segmentation for some of the tomograms. It can be observed that our unsupervised method performs on par with expensive supervised alternative and closely resembles the ground truth. Similar phenomenon can be observed for macromolecule localization, where our unsupervised method even performs much better than the supervised alternative with the same amount of ground truth labels. It also better resembles the ground truth than the unsupervised alternative. We further performed quantitative comparison against alternative supervised and general-purpose unsupervised methods on this dataset. Our quantitative result shows that the membrane segmentation performance of our unsupervised approach lies within 6% of supervised alternative and is substantially superior to the segmentation by general-purpose unsupervised approaches, or SAM. For macromolecule localization, our unsupervised approach performs superior to the supervised alternative with similar amount of supervision. We also performed ablation analysis that underscores the contribution of each component of our pipeline. Being an unsupervised method, our pipeline also generalizes across any cellular tomogram dataset, across different subcellular objects, cells, and organisms. We have segmented membranes and localized macromolecules in C. elegans tomograms using our pipeline, which shows visually reasonable results. We also applied our pipeline to segment membrane-like objects in human RPE cell tomograms. Here, our method could automatically segment actin filaments, which the supervised models trained on annotated cellular tomograms of different cell lines could not identify. All of these shows the high potential of our method in automated discovery of subcellular objects inside the cell. Thank you for watching the presentation. You can find the GitHub repository of our project in the QR code. This work was supported by USNIHR 35 grant and NSFDBI grant. If you have any questions, please email us at mru@andrew.cmu.edu. Thanks.